Hello everybody, my name is Jiawei. I am here to share with you my honors thesis project. As you are watching this video, you're breathing and are alive because your body cells are kept alive by constantly dividing. The division and growth of human body cells are strictly controlled by the cell cycle. Cancer results when normal cell cycle checkpoints fail and cell growth is not in control under normal circumstances. All living organisms rely on cell growth to carry out their generation. Cell growth and development in turn requires efficient duplication of the genome to create daughter cells that contain the same genetic information as the parental cell. The process of duplicating the entire genome is called DNA replication. When this process is disrupted, cells experience DNA damage that leads to genome instability, which can lead to cancer. My project focused on the role of a protein called MCM10, which stands for Mini Chromosome Maintenance Protein 10. MCM10 is an essential replication protein, meaning that without the presence of MCM10, a cell will die because it fails to initiate DNA replication. It is a protein that is part of the DNA replication machinery complex, which is in turn responsible for duplicating DNA with high fidelity. Recent data from our lab showed that human colorectal cancer cells overexpress the protein MCM10 by 2 to 3 folds than non-cancerous human retinal epithelial cells. Additional data suggests that reduction of MCM10 by half can significantly decrease the viability of human cancer cells. Based on these data, we are testing the hypothesis that cancer cells are dependent on high level of MCM10. To fully test the effect of MCM10 on cancer cells in comparison to non-cancerous human retinal epithelial cells, my project aimed at creating a mutant human cancer cell line that contains only one copy of the protein. This way, the mutant cells will make half the amount of MCM10 protein in comparison to the wild-type protein. We will obtain the cell line by knocking out MCM10 genes using the latest CRISPR-Cas9 targeting system. Once we have the mutant heterozygous MCM10 cell lines, we expect them to express half the amount of MCM10 as compared to the wild type. We can then compare the proliferation rates and the cell's viability in response to genotoxic drugs such as hydroxyurea between the mutant cell lines that produces half the amount of MCM10 with the wild-type cells. After comparing that, these experiments will define the consequences of MCM10 deficiency in human cell lines. If cancer cells, not normal cells, are addicted to high levels of MCM10, then this protein could potentially become a novel cancer therapeutic target. Well, that goal is along the way after a series of other research projects. My project is one of the small steps in between here to establish the relationships between cancer cells and their dependence on MCM10. Well, that is the main goal in the construct of my honors thesis project. Thank you very much for listening. Have a good one.